Welcome students. Here in the last class we have seen a deviation from Mendel's principle, and that was incomplete dominance, where a gene was unable to dominate completely, and because of which we were getting a blended phenotype, that is mixed phenotype. We were getting a mixed phenotype, that is in Mirabilis jalpa and snapdog, that is dog flower. Here what we were getting is, instead of red color, the dominating one in F1 generation, we were getting pink color, because red was unable to dominate completely. It was showing dominance, that is why we are getting the pink color, but it was unable to dominate completely. It was able to dominate partly, it was unable to dominate completely. So that is why instead of red we were getting the pink phenotype. We were getting the pink phenotype. Here today we will also see one more deviation from the Mendel's principle, that is we will see codominance. Another deviation here is codominance. Co means together. Here both the characters show dominance. According to Mendel, only one character will be dominating and other character will not be able to dominate. But here in this deviation, in this variation from Mendel, here both the characters are able to dominate at the same time. Both of them are expressing. In Mendel's, only one character was expressed, only one parental character was expressed in F1 generation. But here in this variation, both of the characters are able to express themselves. In codominance, what we see? Both characters, both traits express themselves. Express themselves. Let us see what is this codominance by taking the examples. Here in this, the first example is coat color in cattle. The first example which shows codominance is coat color in cattle. So here we have the coat color is black color. In cattle we find black color as well as we find white color. So we have crossed two cattle. That is, one cattle was having black color coat and another cattle was having white color coat. And usual black is the dominant one and white is the recessive one. Black is the dominant one and white coat color is the recessive one. So this black and white are crossed. Black and white are crossed. That is, a cattle with black color is crossed with a cattle which is in white color. Small b, I already said you, we don't use white W for representing white. We use the small letter of the dominant character. The dominant is black, so we use capital B, and for recessive we use small b. We don't use W. So here black color and white color cattle are crossed. So these are the parents. These are the parents. Pure line, because here also we start with pure line. Pure homozygous dominant, pure homozygous recessive. Okay. So now when this is crossed, when parents are crossed, parents first will produce gametes. So this parent with black color has produced two gametes, that is capital B and capital B. This two will be together now, they will be separated, gametes will be formed. Capital B and capital B is the gamete formed by this black color cattle. Small b is the gamete formed with the, that is white color cattle. Now in F1 generation, these gametes will be crossed. In F1 generation, these gametes will be crossed. For example, this BB, we take it as male. Black color we take it as male and white color we take it as female. Okay. Female means here this gametes will be egg. Male means the gamete will be sperms. So this B and B is the sperm and this B and B is the egg. Now both egg and this one, this egg and sperm will fuse. This egg and sperm will fuse. So here also we take these gametes on one side, these gametes on male gametes on one side and female gametes on another side. Male gametes on one side and another female, that is female gametes on another side. So when we cross it, so here it will be capital B, small b. Here also capital B and small b. How we are getting? We already know. This B and B, again this B with this B. Then here this B with this B with this. So what we get is all four capital B and small b. All four of them are capital B and small b. That is 4:0. All four of them are heterozygous. All four of them are heterozygous because it is capital B and small b. So that means they are heterozygous and all four of them. So according to Mendel, in F1 generation, because this is the parent, these are the gametes, when these gametes have been crossed, what we get is the F1 generation. This we get is the F1 generation. According to Mendel, all four offspring should be of black color. All four offspring should be of black color because dominant traits will be expressed. Which is the dominant one? Black color. So F1 में कौन सा express होना था? Which should have been expressed in F1? Black color. But here instead of black color in F1 generation, what we are getting is white coat with black patches. We are getting white coat with black patches. Here white is also expressed. White color gene is also expressing and black color gene is also expressing. We don't have complete black coat here. In F1 generation, according to Mendel, this should, because capital B and small b, this should have been black. This should also have been black. This should also have been black. This should also have been black according to Mendel in F1 generation. But it is not black. It is having white coat. There is no black coat. It is having white coat and black patches. It is not that black is not expressing, white is expressing, white coat aaya to white is expressing. It is not like that. White is also expressing, black is also expressing. According to Mendel, in first generation, only black should have been expressed. White should not have been expressed. White should not have been expressed as white is recessive. It will be, white will be suppressed by black color. Black color white color को दबा देता. Black color will be suppressed by white color according to Mendel. But here none of the characters are getting suppressed. Both of them are getting expressed. According to Mendel, only one character will express and other will get suppressed. That is, black will express and white will get suppressed. But here in this case, black is also expressing as well as white is also expressing. So white, how white is expressing itself? The color of the coat is white and the coat is not completely white. On the white color coat we have black color patches. On the white color coat we have black color patches. You might have seen the sheep's, cattle, you might have seen the cattle, all black color cattle, all white color cattle, and some cattle shows white color with black patches, some will show black color with white patches. So that black and white patches is because of the codominance, that is both the traits. Here which trait we are using? Color. Both the colors are expressing. None of the color is suppressing. Neither black is suppressing itself or neither white is suppressing. Black is not able to suppress white, white is not able to suppress black. So when both of them are unable to suppress each other, both of them are getting expressed. दोनों भी दिखाई दे रहे हैं, both of them are getting expressed. So this type is called as codominance. Co means side by side, along, one along the other. Both of them are expressing. Mendel में only one will be expressed, another will not be expressed. So we get the parental type only in the next generation. Parental type characters only in the next generation. Variation was, first we have seen incomplete dominance, where we were getting intermediate type. In incomplete dominance, where a gene was unable to completely dominate. Unable to completely dominate, so we were getting the mixed phenotype. Now here, we were getting the intermediate phenotype in incomplete dominance. Here we get mixed. Both of them are getting. White is also there, black is also there. A mixture of black and white we are getting. What we are getting in codominance is a mixture of black and white. So that is, we are getting mixed phenotype. In incomplete dominance we are getting intermediate, neither white, neither red. We are getting pink, intermediate phenotype. Here we are getting mixed. We are getting both black as well as we are getting black as well as we are getting white. So in codominance we get mixed phenotype. In codominance we get mixed phenotype, and in incomplete dominance we get intermediate phenotype. Now this was, we were getting all the four of them are heterozygous, that is 4:0 genotypic ratio, and phenotype all four of them were white color with black patches, white color coat with black patches, that is 4:0 white coat color and black patches, So the genotypic ratio is also 4:0, that is heterozygous. All four are heterozygous, that is capital B and small b, and phenotypic ratio is also all four capital B and small b, that is black patches with white coat color. Now in F2 generation, from first generation we will take any two. From first generation we take any two, that is called as selfing. On selfing we get F2 generation. So I have taken this and this. Okay, this capital B small b and this capital B small b. The gametes which we get is capital B and small b, capital B and small b. So when I put it here, this will be on one side, this will be on another side. So what I get is 1 capital B 1, capital B small b 2. And here this small b and small b 1. 4 now, 1, 2, 3, 4. So 2, 3, 4 we get. So we are getting 3 genotypes. First genotype is both capital B. Second genotype is both small b. And the third genotype is capital B and small b. So if this is both BB, which color it will be? Black color coat. Small b means white color. Capital B and small b means black color, because B is dominant now? No, we get black patches on white color coat. Black patches on white color coat. So what we get is mixed phenotype. We get is mixed phenotype. So according to phenotype we get the ratio 1:2:1. 1 black, 1 white, 2 is mixed one. 1 black, 1 white, 2 is the mixed one. So genotypic ratio also 1 capital B, 1 capital B small b, 2 capital B small b and 1 small b. So phenotypic ratio and genotypic ratio is same in both the generations. In F2 generation, the both phenotypic and genotypic ratios are the same. In codominance also, in incomplete dominance also, both genotypic and phenotypic ratios are same. But the difference in codominance and incomplete dominance is, in incomplete dominance, a gene will unable to completely dominate. So that is why we get intermediate phenotype. And in codominance, both the genes are able to dominate. So that is why we get mixed. Both means mixed phenotype in codominance. So this was one example which shows codominance. We have another example which shows codominance here in this. I'll erase this one. So here, see from here. This is another example, that is size of lentil seeds. Lentil seeds. One is spotted lentils. On the lentils, for example, this is the lentil, big spots was there. Big spots. Big spots, irregular spot was there, patches was there. And on this another lentil, like this, small dots, dotted patches, small patches was there. So a plant with dotted lentils, a plant with spotted lentils. A plant with spotted lentils, that is patches, big patches, a plant with small patches. Okay, so both of them were crossed. These are the parents, that is a plant with spotted lentils. For each gene we have two alleles. Now this is one allele, this is one allele, homozygous alleles, that is spotted lentils. Then this is the plant with dotted lentils. We have seen now we use TT for height, 2T. So here also CD, because two alleles, each gene will have two alleles. So here spotted lentils and dotted lentils were crossed. So gametes from this are the gametes. So this one is one gamete, this one is another gamete, this is one gamete, this is another gamete. When these gametes are put on one side, this on one side and this on one side, in F1 generation, we get all four of them are CSCD. All four of them are CSNCD. Now which color, which it should have been? This one is the big and regular patches is the dominant one. This is the recessive one. So in F1 generation, what should we have got? We should have got all the lentils with big patches. We should have got all the lentils with big patches. This is F1 generation. Now all should be dominant. Dominant means we should have get all the spotted lentils. We should have get all the spotted lentils according to Mendel's law. But here, even the CS and CD, even this is dominating gene, we are not getting spotted lentils. We are not getting spotted lentils. Instead we are getting both spots as well as dots. We are getting lentils on which shows both spots as well as dots. We are not getting this, we are not getting this. Instead we are getting lentils which are spotted as well as dotted. We are getting lentils which are spotted as well as dotted. So the CS and CD are nothing but lentils with both spots and dots on them. So this is the mixed one. We are getting this as well as we are getting this one, mixed. In your textbook this example is given and this example is given. So this is in F1 generation we got is all four of them are heterozygous, that is CSCD. Heterozygous means 4:0. All four of them are this heterozygous only. So that is 4:0. There is no homozygous. And all four of them are spotted and dotted. All four of them are spotted as well as dotted. So it's for phenotypic ratio is also 4:0. So this one, if you do it in F2 generation, so in F1 generation what we got is this 4 offsprings. So out of this 4, 2 we will do, cross it. So one CSNCD, this one, and another CSNCD, this one. So we are crossing. So gametes we get CSCD, CSCD. This will be separated. That's it. So gametes we get. Put the gametes here. What we get is both this and this means CS, CS. This and this means CS, CD. This and this means CS, CD. This and this means CD, CD. So what we get is, one is this one, CS, CS is 1. Then we get CS, CD 2, because this is also CS, CD, this is also CS, CD. And the third we get is CD, CD 1. So one is spotted lentils, one is dotted lentils, and another is spotted and dotted. Spotted and dotted lentils. So here also the ratio is the same. 1:2, 2:1 is the phenotypic ratio, that is spotted, dotted and dotted, and genotypic ratio is also 1:2, 2:1. Genotypic means 1 both CS, CS, 1 both CD, CD, and 2 CS and CD. So the genotypic ratio and phenotypic ratio is the same. So these two examples are the examples which shows codominance, in which both the genes are able to express itself. Both the genes are able to express itself, and we are getting a mixed phenotype. We are getting a mixed phenotype which is seen in, we are getting a mixed phenotype, and both the genotypic ratio and phenotypic ratio are the same. So these were the variations from Mendel's principles. In the next class, here one more example is there, blood grouping in humans. It is also an example for codominance. We have already seen 2 examples. Now we will move on to another topic, that is pleiotropy, pleiotropic genes. What do you mean by pleiotropy and what are that is, we will see in the next class.